Um, so I'm very, very pleased to introduce our keynote speaker for this conference session. Our keynote speaker today is Dr. Stephen Gray. Dr. Gray, I believe you are here and your audio and video will be enabled. While you pull up your slides, I will do a brief introduction for you. So to give you some background on Dr. Gray, he is with UT Southwestern. We've had a conversation and a relationship with Dr. Gray for quite some years now. He's been very supportive of the efforts of the ACD board as well as on the Gene Therapy Consortium. We're excited to have Dr. Gray speak today because he is at the forefront of several researchers advancing to clinical trials with gene therapies for rare diseases. Today he's going to talk about AAV-mediated gene therapy for neurological disorders. This is particularly pertinent for our community because both CTD as well as GAMT have significant brain involvement. So any gene therapy that would go to the clinic, we'd hope for it to actually rescue the disease in the brain as well. And I think there's some nuances to the types of gene therapies that work all across the body versus what works in the brain that I'm hoping that Dr. Gray can educate us on. So with that, Steve, I'll let you take it away. I'm not sure if you're muted, but we are not able to hear you.
Yep, just took me a second there to get get everything set. Yeah, it's a real pleasure to have been invited to this, the CCDS conference, and I really had not been in touch or with this community at all until really right before COVID. I think the fall before that, with the last in-person meeting that I was able to attend. But I think I was invited, and I'm happy to speak today just about what our lab has been doing and kind of what's been happening in the field around gene transfer to the central nervous system. And so, as with the introduction, I'm Stephen Gray. I'm an associate professor at University of Texas Southwestern Medical Center in Dallas. And I'm, let's see, second, okay. Really quick, I've got some disclosures because my lab does a lot of work developing AAV vectors and developing treatments for different neurological disorders. Many of those get licensed to various companies that have created various conflicts of interest to me personally. So I'll start off with an introduction to AAV, which I think, everybody's been talking about this, but just to make sure that everybody's on the same page right at the beginning of my talk, this is adeno-associated virus. It was discovered, about over 50 years ago as kind of a contaminant of adenovirus stocks. And it was basically studied and developed and eventually viewed as potentially a good vector for gene therapy to deliver therapeutic genes, because essentially you can remove all of the viral coding sequence, gene coding sequence, and replace it with any DNA cargo that you want it to carry. And it's packaged within a very stable capsid. It's about 25 nanometers. And there's a few natural features of the virus that make it attractive. One is that the virus itself is not associated with any pathogenicity in humans, so it's essentially a safe virus just in nature. But you have many, at this point, hundreds of different variants of the capsid available that can create this toolkit of some of these capsids that'll direct the gene to the liver, some to the muscle, some to heart, and some to brain. And there's now been, literally hundreds of clinical trials using AAV. It has overall a very safe track record, although in the last year there have been some very significant adverse events with AAV, including the death of some patients in trials that use very high doses. But the vector itself has been now achieved full regulatory approval in Europe and the US for some AAV-based products. So there's a track record now of these being developed past the clinical trial stage and actually to fully approved drugs. I'm going to introduce, and my talk is really going to be focused on delivering AAV and trying to treat nervous system disorders. So I wanted to start off and pay a little bit of a tribute to a study that I'd say really made everybody in the field aware of the possibility of doing gene therapy for neurological diseases. And this is for a disease called AADC Deficiency, where patients are not able to make dopamine. It's an inherited disorder, a gene deficiency, very rare. But the quality of life, of this disease is incredibly poor. The prognosis of these patients are poor because they can't make dopamine. So it's really, they're left in a situation where they can't really make purposeful movements. And so you can imagine that, this is a devastating disease with early death. But there was an approach at the time of trying to deliver the AADC gene to treat Parkinson's disease, to try to allow Parkinson's patients to make more dopamine. And there was an investigator, physician scientist in Taiwan that took this vector, this essentially Parkinson's vector design, and had patients with AADC deficiency that they administered this. And this was administered by intracranial administration. So they drilled burr holes in the skull, inserted a cannula to target the thalamus, and then administered this bilaterally directly into the brain. And I think you can see in this video a very transformative benefit to patients that received this therapy. They were not cured, but it was a very striking change and a very striking improvement in their lives. And some of these patients, especially those that were treated early, were able to start, having, certainly a better life, but also even going to school and starting to develop. So this treatment has been administered, to dozens of patients in Taiwan and then Japan. This trial has moved to the US, and then a new iteration of this trial has been started by a physician scientist named Christoph Benkiewicz at Ohio State. But my point of showing this is that I think this was the first instance that I can point to of really unequivocal benefit being seen from a gene therapy that targeted the nervous system. Of course, this was all happening, and this was almost a decade ago now, but this utilized this approach of stereotactic administration of the vector. It worked for this particular disease because the population of dopaminergic neurons were localized to a specific area of the brain, and you could deliver the gene straight to that small area and impart a very profound benefit. Again, that was specific to that disease. For most diseases that we're talking about, they're gene deficiencies that involve the brain, we want to deliver the gene broadly across the entire brain. And there's still some approaches that are trying to use intracranial administration, but this is an approach that can look really fantastic in a mouse where the brain is very small, and you can fill much of the brain volume of a mouse with the gene. But if you scale that to humans, you end up with each injection kind of targeting and treating something the size of a marble, and you really can't do enough injections or spread them out enough to really comprehensively treat the brain in a human. So my lab, along with other labs in the field, started to pursue other ways to try to broadly target a gene across the brain. And one thing is that there's some vectors, some capsids that were identified that you can inject IV, and a portion of those will get across the blood-brain barrier and get into the brain. Or something that we settled on was doing intrathecal administration of the vector, so essentially administering it via spinal tap into the spinal fluid, and that fluid is produced in the brain, flows down and bathes the spinal cord, and then flows back up and bathes the brain. So it's kind of a fluid volume that has the potential to transport the vector all throughout the central nervous system, but kind of concentrate it there in a way that you won't get from an IV injection. So going forward, this is work that we published, that my lab published in 2011, of doing tail vein injections in mice. And you can see visually here, these are kind of, cross sections going, rostral to caudal through the mouse brain. And all of the dark staining here is where the GFP transgene was expressed. So you can see that it is, broad, targeting every brain structure. And this was what we kind of quantified in that paper, was it was a mixture of neurons and glia, mostly neurons and astrocytes. So this works, really well in a dose-responsive manner in mice. The more you put in, the more that gets to the brain. But if we look into some of the details of this, then some logistical challenges and maybe some unfavorable components of this exist. One is if you look at where the virus is going across the whole body and don't just focus on the brain, then this is a graph that's quantifying how many copies of the genome go per cell in different tissues. So you can see the brain and the spinal cord here on the graph. And then you see larger amounts of the vector are actually going to muscle, heart, kidney, spleen. And if you look at the difference in the y-axis scale, it's about 50 to 100 times more vector goes to the liver as it does to the brain. So we did pursue this further and kind of did a comparative study, injecting the same vector into mice and then also injecting it into a number of non-human primates. And overall, our conclusions were that this can work very well to target the brain. And the more you administer, the more gets there. But when we translated it to non-human primates, overall in our hands, we saw the efficiency of it decrease. In particularly, it decreased in terms of the virus going to neurons. Most of the it started to shift, and most of the virus is going to astrocytes. But overall, for us at the time, we were trying to scale these doses, look at how these doses would scale to humans, and realized it was an enormous amount of vector that we would have to administer and manufacture to humans. We were getting lots and lots of the vector going to peripheral organs, which in some cases can be problematic and can lead to a lot of adverse consequences. And then also about a third of the human population has pre-existing antibodies against AAV9, and even a low level of these antibodies will block the AAV getting to the brain. So we're kind of faced with this paradigm here of, we inject the vector IV and it basically goes everywhere across the body at different levels of efficiency. And so we wanted to see, could we modify the virus to target it more towards the nervous system? Or, the simpler way is to modify the route of administration. And we are doing a lot of work trying to modify the virus, but maybe that's another story for another day. The simpler approach is to see if we could administer the virus differently. And that's where we shifted to doing intrathecal administration of AAV9. And so, this is done, intrathecal injections are done pretty routinely as an outpatient procedure by, most neurologists or anesthesiologists. You can get more complicated with this by injecting the vector higher up, kind of at the base of the skull into the cisterna magna, or doing stereotactic administration into the ventricles, which are kind of the reservoirs of CSF in the middle of the brain. But we really settled on doing just lumbar intrathecal administration. We can model this in mice. You can do lumbar punctures in mice. It's technically difficult, but we got good at it. And I'm going to just show you, this is a slide. This is work that it published with Rachel Bailey, who's an assistant professor at UT Southwestern. We published this last year, which was really a comprehensive mapping of where AAV9 will go after intrathecal administration in mice. We also compared this to cisterna magna. And so you can see visually, this is a GFP vector. You know, the staining we did shows all of the transduced cells in black. So, so basically all of the vector is going, you know, it is still distributing broadly across the brain. It's getting into even deeper brain structures. It is more efficient in the spinal cord. And then if you zoom in just by morphology, you'll see that it's a mixture of neurons and glia, which is similar to what we saw from an IV administration. And we did quantify the amount of the virus that's going to different brain structures and different components of the nervous system, and also peripheral organs. And there's a couple points that I want to make here. One is if we just focus on the nervous system, if you can see the axis on the scale, this is like copies of the gene per mouse genome, or copies of the gene per cell. And so in the spinal cord, especially the lower spinal cord, you know, on average, we're getting 1 to 2 copies of the gene per cell on average. And you look at things like the dorsal root ganglia, same thing. But when you get into the brain, on average, we're getting something more like, you know, 20% or 5% or 20% of the cells that are on average that are receiving the vector. And this was kind of what we considered close to a maximum feasible dose in terms of how much we can concentrate the virus and what volumes were practical. And this is just a real limitation of the technology right now, where you know, we can target the brain with an AAV9 vector, whether it's IV or intrathecal, and we are not going to target every cell in the brain. We're going to target a minority of cells throughout the brain. And I'll show you later that minority of cells that are transduced can still provide a meaningful benefit, but that might depend disease by disease what that looks like. The other point that I wanted to make is if you look at the peripheral organs, you do still see a significant amount of the vector going to peripheral organs, even though we're injecting it into spinal fluid. In fact, we're seeing more vector going to the liver than we are to the spine or to the spinal cord where we're injecting it. And we've done some kinetic studies showing that, you know, in fact, a good portion of the vector, you know, if I had to put a number on it, maybe a third or maybe as much as half does escape and get into the blood. So this could be good or bad depending on your application. But in many cases where it's you know, the gene has functions across the whole body, I would view this as a positive because you can do one intrathecal administration and then you also get the benefits from a lower dose IV administration. So this is all in mice. We did studies in collaboration with a neurosurgeon at Emory University named Nick Boulis, and we administered this vector into farm pigs, about 15 kilogram farm pigs. And we published the spinal cord, you know, delivery in 2011. We did not include in that publication, you know, what the transduction looked like in the brains, but we did end up getting those and we did histology. And so as I'm showing you here, we still got pretty extensive gene transfer, you know, to the brains, and particularly, you know, very high transduction of Purkinje neurons in the cerebellum. We did advance this and test it in mice, or sorry, in non-human primates. We published work on this in 2013, and this has been extensively published by other groups as well, where, you know, what our conclusions were that is that intrathecal administration of AAV9 can still lead to widespread vector distribution across the nervous system in primates. And in our view, intrathecal administration overcame many of the barriers we identified from an IV administration, including, we could deliver a lower overall dose to kind of get the same amount of gene transfer to the brain. We could avoid these circulating neutralizing antibodies. So we administered this to some animals that had low levels of antibodies against AAV9. And those antibodies are in the blood, but unless those antibody levels get very high, they don't show up in the spinal fluid. So this was a way to hopefully avoid excluding, a third of the population from being able to receive it. And also, we still got plenty of vector to peripheral organs, but instead of this 100-to-1 ratio of liver to brain, now we're, getting closer to an even distribution between the nervous system and the peripheral organs. So this was all work that we did, essentially to try to explore and develop this general approach for delivering a gene to the CNS. Now, I'm going to take a segue into kind of the why, and I'm sure the why is very intuitive for everybody, but for me particularly, I got started on this because of a disease called giant axonal neuropathy, and it was really driven by, one parent-driven foundation called Hannah's Hope. And so this is Lori and Matt Sames. Their daughter was diagnosed with this disease when she was 3 years old. As I'll go into, this is a terminal disease. It's fatal by around 20 years old. But they went on the journey that I think many of you are familiar with of trying to drive research and drive the development of therapies. So this was my personal inspiration when I got started to try to, develop this approach. And when they came to us, there was no mechanism to deliver the gene for GAN in a way that would be therapeutic. This was all work that was developed, really, driven, inspired, and in many cases funded by Hannah's Hope. So this disease, giant axonal neuropathy, it's an autosomal recessive disorder. It is a degenerative disorder, and you can kind of see the visual progression of the disease here with, first at a young age, clumsiness that leads to a loss of ambulation, which, eventually leads to something close to quadriplegia with respiratory distress. And as I said, it's typically fatal by around 20 years old. It gets its name. It's called, like I said, it's autosomal recessive disorder. It's caused by a lack of a gene called gigaxinin, and it's called giant axonal neuropathy because when you're missing gigaxinin, you get this accumulation and disorganization of a variety of intermediate filaments, and it gets its name giant axonal neuropathy because if you were to do a sural nerve biopsy in a young GAN patient, then you would see these large distended axons that are densely filled with neurofilaments. So, our approach, again, all of this work trying to explore AAV9, explore ways to deliver a gene to the brain and spinal cord, it was all focused around GAN. So as we developed this approach and applied it to GAN, then we packaged the gigaxinin coding sequence with a very minimal promoter into a self-complementary AAV genome, packaged it in the AAV9 capsid, and did a variety of proof of concept studies and safety studies that eventually led to a clinical trial for this disorder. When we started with GAN, it was really thought of as a quote unquote motor neuron disease. As we've studied this disease, it is much more complicated than that, but our primary targets were still the spinal cord and brainstem, motor and sensory neurons, but the more vector that we can get to treat aspects of the brain disease, the better. So if you're curious, I'm not going to go through all the preclinical data. This isn't really a symposium on GAN, but if you're curious, you can look up the paper that we published last year in Molecular Therapy on all the preclinical studies that really drove the efficacy for this. Importantly, we partially corrected a motor phenotype in the mice. We corrected many aspects of the pathology in kind of key areas like, peripheral nerve and dorsal root ganglia. And importantly with this, we did comprehensive GLP toxicology studies in 120 mice, 14 monkeys, and 60 rats. And this had a very favorable safety profile. And I should note, for this treatment, we did not see any toxicity in the dorsal root ganglia, which has been reported for a number of AAV intrathecal approaches. But that may be because we used a minimal promoter in this application. So this is kind of a bird's eye view of what this looked like. In 3 years from the start of funding from Hannah's Hope, we got what I'll call good lab results. This is where we did, explored and developed this approach using intrathecal AAV9. And this is where we did proof of concept studies in cell culture models and also the mouse model of the disease, and ultimately went to the FDA in January of 2012 to propose doing a clinical trial for this disorder. And that meeting actually went very well. I'm not sure if anybody had proposed an intrathecal gene therapy trial to the FDA before, but they were quite receptive, quite encouraging and supportive. So they more or less said, yes, go ahead and go forward, but do it responsibly. Do it responsibly for the FDA means, do fairly extensive toxicology studies, pharmacokinetic, pharmacodynamic studies, clinical trial design, and then go through the alphabet soup of regulatory agencies. So it ended up being about 2015, about 6.5 years from when we started, when we were able to initiate this phase 1 clinical trial. And I should say that this, again, acknowledge Hannah's Hope because they funded this pretty much 100% up until the start of the clinical trial. I'll take a minute to acknowledge my collaborator in this, my clinical collaborator, Karsten Bonneman, who's at the at NINDS. They took this trial on, they reviewed all of our preclinical data and were essentially willing to run this clinical trial, for free and with all of the expertise that they could bring to bear on it. And Karsten has been an incredibly good collaborator and in many ways a mentor. So we hit this milestone in May of 2015 of the first in human intrathecal gene transfer to broadly treat a CNS disorder. And as far as I know, this is for any disease. And it happened for this very rare disease that nobody had ever heard of and nobody really ever cared about. And it really happened because of this woman here, Lori Sames, who was and still is an incredible patient advocate that really drove all of this. So an overview of this clinical trial, like I said, we packaged the gene with minimal promoter within an AAV9 capsid. We initially designed this as a single dose clinical trial, but even as we started the clinical trial, we were still doing preclinical studies and quickly gathered data that a higher dose could be even more beneficial. So this turned into a dose escalation trial. And as an academic investigator-initiated trial, we had a lot of flexibility to really look at the data as we were moving along and make any adjustments to this that we needed to. So it was kept at a constant infusion volume. We formulated the vector in a buffer that was heavier than the spinal fluid. So to help with the physical distribution of the vector, towards the upper spinal cord and brain, we kept the patients in a head down Trendelenburg position for about an hour post-injection. And to date, we've treated 14 GAN patients in this clinical trial. I wanted to touch on some immunology aspects, and this could be very, very relevant for any of the CTDs. So in At this point, some of the clinical trials we're proposing, and actually one that we have ongoing at UT Southwestern, we're administering 1 × 10⁻¹5 vector genomes in a patient. So it's 1 quadrillion virus particles, not including the empty capsids. The immune system will see that. And there's 2 immune concerns that we can think about. One is any kind of very rapid responses against the capsid while it's around and the viral particles themselves. And so we want to blunt any kind of initial acute immune responses. But then also, we had some patients we wanted to enroll in the trial that had basically total biallelic null mutations, so they don't express the protein at all. And those patients we would call cross-reactive immune material negative or CRIM negative. And another way of saying that is their body has never seen gigaxinin. So if we administered this gene therapy, their immune system may react and reject the therapeutic protein that's being expressed. So this evolved over time. It started off with a simple just 30 days of steroids. This evolved into doing 4 months of steroids. And it also involved in some additional T-cell suppression with a drug called rapamycin or sirolimus. And then for the patients that we would be worried about them mounting an immune response against our gene, we also put them on an additional T-cell suppressant, tacrolimus. And then we monitored these immune responses very carefully throughout the trial. And I should say that all of these patients developed very high levels of AAV9 neutralizing antibodies. So we've very effectively immunized them against receiving a second dose. And some of them have mounted T-cell responses against the capsid that probably come too late to have any consequence. But importantly, none of the patients have developed a immune response against the therapeutic gene, which is really what we were trying to accomplish critically. And overall, the safety of this has been quite good, the safety of the trial overall. It's been well tolerated across all 14 subjects at all 4 dose levels. I'll touch on the efficacy data a little bit. This is not published, but we benefited for this disease of a natural history study that was being run starting a couple years before, or a few years before the clinical trial, and has run concurrent with the clinical trial. And that natural history study kind of guided the selection of this motor function measures 32 scale, or MFM32. And it was actually across the spectrum of the disease, there was a very consistent 8-point decline on this scale per year for any individual patient. So we had a nice and consistent slope of decline that we could predict for this disease, and then we were able to see with the patients whether they deviated from that slope post-treatment. We could also, many of these patients participate in the natural history study, so we were also able to basically compare each patient on an N of 1 basis to see how their disease progression compared with their lead-in natural history data. So this is just the kind of aggregated average data on the left, where I think you can see some trends where the 1x dose in 2 patients, these are also 2 of the older patients, didn't seem to show much of an effect. But when we move to this 3.3x dose and 1x dose, then there's the appearance of maybe a leveling off where they're not declining. And on the right is just the raw data for each individual patient, so you can see how each of them look. We also have 3 patients dosed at the equivalent of 10x, but the last one of those is just hitting the 1-year time point, so we don't have data on that group to report yet. We did incorporate some fairly sophisticated statistical analyses on this, and one of them is a Bayesian analysis, which I am not an expert on statistics, but this is a type of analysis that's particularly suited to rare diseases where there's small patient populations, maybe looking at large effects. And here, again, we see sort of the same data plotted, just the averages without the error bars. And then we have some p-values here that are reported where the deviation from natural history and at least some slowing of decline was highly significant, even with these small numbers of patients. So, overall, this is still a work in progress, but I think we're encouraged by the data thus far. We're following all these patients out long term. And as I said, we're anxious to see what the higher dose looks like. So I was going to just maybe segue off again and talk about this as a platform type of technology or platform approach that could be applied to other disorders. And, and first start off with this question of how does this happen for such a rare disease? And I again, acknowledge Lori Sames and her work with patient advocacy is really driving all of this forward, corralling scientists, but also a committed, supportive community of volunteers that frankly raised a lot of money. And the number that they'll put out there is they raised about $6 million in 6 years. Now, they were funding a lot of different areas of research, not just gene therapy. So that wasn't the price tag for the gene therapy, but it's basically something where anything that we wanted to do, anything we wanted to pursue, they found a way to make it happen. But frankly, on their part, it was exhausting. And what this looks like in reality is fundraiser after fundraiser after fundraiser, and an exhaustive community that made this happen. But I mean, they're really burnt out now. So what are the magic ingredients? And this is just my perspective, is I'd say link scientists and families. This is what's happening right here in this meeting. I think that you have to have compassion, but compassion doesn't work without know-how. We have to have a good approach. You have to have accountability, and on our end, just knowing the patients and knowing the stakes of moving things forward quickly was the accountability. And then also, all of this is expensive, so the fundraising component of it can't be understated. And again, I'll just continue on this segue a little bit, and I'm preaching to the choir here. But when we talk about rare diseases, this is a large but I think underappreciated problem. Again, I think you're probably familiar with this, but there are over 7,000 rare diseases. A significantly large portion of the population will have one of these rare diseases. If you look at this in aggregate, 80% of those are genetic, so gene therapy makes a lot of sense for many of them. But this gets really difficult when you think about 50% of these patients are children, and 30% of those children don't live to see their 5th birthday. So you start to see how big of a problem this is, especially considering less than 5% of rare diseases have FDA-approved treatments. Again, I know with this meeting, I'm preaching to the choir, but just in case anybody hasn't really thought about the scope of this, here it is. And just taking that a little bit further, because I like analytics, the Every Life Foundation commissioned a very useful study that was published earlier this year to really try to come up with a number. What's the economic burden of rare diseases in just the US? And this was just for 1 year. And the number they came up with was close to $1 trillion. And about half of that was direct medical costs. And then, but then if you factor in a lot of things that, people would understand, like missing work, having to take care of your child or yourself, forced retirement, a lot of healthcare that's not covered by insurance. This becomes a staggering number. And they actually compared this to costs for things like cancer and kidney disease. And the cost of rare diseases in the US actually dwarfed the cost for some of these other very expensive categories of diseases that we think about. So now, talking about the platform technology, this is a big problem. How do you solve it? I think the NIH is trying to figure this out. How do you tackle 7,000 rare diseases? And it's not a good answer, but what we've been doing is one disease at a time. And hopefully there can be a lot of other labs that are doing what my lab is doing that can also tackle one disease at a time. And then maybe in aggregate, we can start to make an impact. So thinking about AAV9 as a platform technology, again, this is just a non-comprehensive list of kind of the evolution and start of an increasing number of clinical trials. You'll see that most of these are for lysosomal storage disorders, except for things like spinal muscular atrophy, which became FDA approved, and things like Duchenne muscular dystrophy, where IV approaches for this can treat muscle disorders. In my own lab alone, this is a non-comprehensive view of the different diseases that we're going after using this same approach. All of these are being treated essentially the same way. And GAN really led the charge on this, and that's been in a clinical trial for a while. But then we also have three lysosomal storage diseases, Tay-Sachs and Sandhoff disease, that trial is enrolling. CLN7 Batten disease, I'll touch on, that we're enrolling patients at UT Southwestern, and CLN1 Batten that hopefully will be enrolling patients soon. And then we've published on many of these, some not, kind of where we've generated very positive proof of concept results in various animal models, from mice to dogs to sheep. And we're working to try to move these into clinical trials, and then a number of other disorders that I consider at a discovery where we think there might be some applicability of gene therapy, and we're exploring that right now. And maybe I'll skip past kind of the primer on lysosomal storage disorders, except just to say these are conceptually much easier to treat, because you can treat one cell and then it can become a factory to produce an enzyme that is secreted and taken up by neighboring cells. So whenever we talk about this 5 or 10% of cells or whatever that we're going to target in the brain, those cells that directly get the gene could end up treating all cells. And one example of that is a very rare disease called Aspartial Glucosaminaria. And this is just, we published this this year in Molecular Therapy, so you can look it up if you want. But it's basically, we administered these mice either IV or intrathecally. Both worked, but we could get this super physiological level of enzyme that's circulating in the blood for the life of the animal. And so you can see the normal mice in black, the knockout mice in gray, and then our treatment groups at different ages. So really expressed a lot of enzyme. This is in the blood. It was actually lower amounts in the nervous system. But if we looked at there's a toxic substrate that accumulates in this disorder, and you can see it accumulate in the knockout mice, in the blood, in the brain, and in the spinal fluid. And then we see this significant clearance that's dose responsive, in these different targets. And again, the IV approach worked, the intrathecal approach worked, but we focused on the intrathecal approach going forward because I think it just ends up being more practical and using lower doses overall. I was also going to touch on, this is a really, really busy slide, but this is for infantile Batten disease, another lysosomal storage disorder. And there's a lot of data on this slide. We looked at, different doses, combinations of intrathecal and IV, and, ages of administration. And the important point here, these are survival plots, is that if we administer this treatment to mice early, actually before the onset of, kind of severe overt symptoms, the treatment looked fantastic. We got normalized lifespan of the mice if they were treated very early, like a week old. And then as we treated the mice older and older, even before they started having overt symptoms, then the treatment benefit got lower and lower. We did see a treatment benefit of treating mice after they had symptoms, but it was, it was much more modest compared to if we could intervene very early. And for degenerative disorders, I think that this is going to be a consistent theme. We certainly see it across the different degenerative disorders that we work on. This is a different form of Batten disease, CLN5. We benefit from a sheep model, a naturally occurring sheep model of this disease that's in New Zealand. So we shipped our virus to New Zealand and they treated their sheep. And this is looking at the left is a clinical rating scale for that's specific to these sheep, where sheep, and then you see this different areas of slight decline, but overall, fairly stable, depending on when the sheep were treated at 3 months old, 7 months old, 9 months old. And then in red are just the untreated sheep that decline very rapidly and have to be euthanized about 2 years old. But you'll note that, even treating at, 7 months old, that we had sheep living out 4 years and still going. And then we looked at brain volume, did MRIs on the sheep, and you basically have a sharp decrease in brain volume in red in the untreated sheep. But wherever we dosed the sheep, if we treated them early, the brain continued to grow. If we treated them late, then it basically just stopped the disease progression wherever they were at. And this is multiple sulfatase deficiency, another disorder that we collaborate with Jackson Laboratories on, where these mice have a median lifespan of about 11 days. We actually treated them at 7 days old, and I was actually surprised about this result, but treating them at 7 days old when they normally die at 11, we got over 50% of the mice living out long term. And I was going to, just give a little vignette of 2 other disorders before I wrap up here. One is Krabbe disease. This is globoid cell leukodystrophy, another lysosomal storage disorder. Very rare, very aggressive, death by, say 1 to 3 years old. People have heard of Krabbe disease because Jim Kelly, Hall of Fame quarterback for the Buffalo Bills, his son Hunter died of this disease. They've done a lot of work advocating for this, and they've actually got newborn screening in place in a handful of states for this disease. But we benefited from, we published work on mice. The studies in mice are kind of boring compared to the studies in dogs because we had a availability of a dog model of this disease. And those dogs recapitulate most aspects of the human disease. They have to be euthanized by about 16 weeks old. And so we treated dogs very early at 2 weeks old, or later when they started having symptoms at 6 weeks old. And in this case, you can see in black the untreated dogs that have to be euthanized around 16 weeks. Treatment at a low dose very early had some benefit. Treatment of a high dose later had some benefit, but it was really these dogs that were given the high dose very early at 2 weeks old. The paper was published last year at 150 weeks, about 3 years, and those dogs are still going. And so what this looks like is, dogs were given effectively placebo. And then this is where you see kind of the natural progression of the disease in the dogs, where they start to lose motor function, they start to lose hearing, eventually lose eyesight. So this is typical progression and what it looks like at 10 weeks old. This is, one of our treated dogs. It received the early injection, high dose at 70 weeks old. This is all online, it's in the publication. All the dogs looked like this, where couldn't see anything clinically wrong with them. And I think there were some very minor lesions that could be picked up by MRI, and we don't know if those might have already been present at 2 weeks old. Now I'm going to end on one thing, because all of these are these lysosomal storage disorders that can benefit from cross correction. These are like the low-hanging fruit of gene therapy, so the results do look great. But this is another form of Batten disease, and this one is CLN7, and the protein MFSD8 is membrane bound. So this particular disease does not benefit from cross correction. And we treated these mice at 7 to 10 days old, or at 120 days old, again with sort of a highest possible dose or a lower dose. And you can see, in the dashed black line are the vehicle injected knockout mice. And if we treat them with a high dose later, which is in red, all right, high dose later, which is in red here, some benefit, and then the high dose when we give it at 7 days old, a more profound benefit. And we have behavioral data that corresponds with this, that they maintain motor function and strength, throughout their lives, until they succumb to this disorder. But it's definitely not a cure, but it was a substantial benefit. And we actually opened an IND at the end of 2020 for this disease, and we've enrolled 2 subjects at UT Southwestern. The highest got a dose of 1 × 10 to the 15th vector genomes injected intrathecally, and so far everything's been tolerated well. So my overall conclusions are, I guess AAV9, and there are other flavors of AAV vectors, but I think AAV9 has become the sort of vector du jour for CNS gene transfer. Administered either IV or intrathecally has been validated across numerous labs for disease-modifying CNS gene transfer across several species, including humans. But, and this is a big but, as I've said, the CNS gene transfer with AAV9 still targets a minority of cells in the brain. We should not think about this as a cure. We should think about this as a potential treatment. And that's going to vary from one disease to another about whether, treating, say, 5 or 10 or 20% of cells is going to make a big difference. So I'd say in some cases it can mediate a transformative benefit, but for others it may not be enough. And as we've seen in the GAN trial, all the patients we've dosed have been very effectively immunized against AAV9 for years out. So I think that right now we have to assume that these are one-time treatments. So I'd say, not one size fits all, but we might think about this as one size fits many. And so thanks for your attention. I'll acknowledge my team. They work really hard every day to make me look good when I'm presenting all of our work, and have a lot of collaborators in different funding agencies because we've worked on many, many diseases across the years. And with that, sorry if I ran just a hair long, but I'm happy to take questions.
Thanks, Steve. So we definitely have questions in the Q&A box for you. I've also invited some of our previous speakers so that they may participate in closing out this session, make it more of a discussion. Just for those of you who were brought up as panelists in the past hour, please feel free to chime in with questions. I'm happy to start with questions from the Q&A box, and I'll pause after every question to make sure that if our panelists have something to say, then they have an opportunity to do so. Our first question, Steve, is how relevant is the severity of a disease for the FDA to allow gene therapy trials, depending upon whether a disease is fatal, non-fatal? Are there different bars?
Well, I think everything has to be a risk versus benefit. And I think that the severity of the disease can inform, basically what's the risk of doing nothing. And I think that, at the end of the day, you don't get a pass just because it's a really, really severe and early fatal disease. You still have to meet the bar of having safety and having, really achieve that proof of potential benefit to the patients. But certainly I think a very aggressive and fatal and debilitating disorder would get it would probably get a little bit easier path through, just, weighing the risk versus benefits.
I think a corollary, follow-up to that question is for diseases that are non-fatal and non-degenerative, progressively degenerative, is it just harder to initiate a gene therapy clinical trial? And I say this from the vantage point of CTD and GAMT.
Yeah, I mean, it's always, you can kind of talk about, like, everybody talks about their individual experience with the FDA, but, a lot of it is, how do you present the argument? How do you present your rationale for what you want to do? I think there's gene therapy that's moved forward. I mean, look, gene therapy moved forward for hemophilia, which is a devastating disorder, but it had a treatment. It had an effective treatment. It's a difficult treatment. It's a very expensive treatment, but it had a treatment, and multiple hemophilia trials went forward. So I think in terms of where a very devastating, very, early fatal disease may move forward a little bit easier, it may be more of something where, okay, you've got some safety issues, but you've got strong efficacy data. They might say, well, weighing the risk versus benefit, even if we have some, small adverse consequences, it's still a huge net benefit to the patient. But I view it as, if it's something that's not as severe, then, you're really relying on stronger efficacy data and really clean safety data.
Thank you. So I noticed that Steve Baker, one of our previous speakers, had a question. Steve, I believe you're a panelist. Would you like to ask your question to Steve Gray?
Yeah, sure. Thanks. Hey, Steve, that was a fantastic talk. I mean, the number of diseases you guys are working on is super impressive, and really great work. So I guess the one thing I was wondering is, when you're delivering the vector intrathecally in mice, you saw it accumulating in the salivary glands and the esophagus. And I'm wondering, is that at all related? Are the mice co-housed, or are they cleaning themselves after the injection? Is it possible that that's something that you wouldn't see in humans?
Well, I think you probably would, to some extent. I mean, it's possible. To be honest, that was something where we've been looking at delivery to the autonomic nervous system. So we were starting to try to see how much might go to different peripheral autonomic ganglia. And this was kind of a first pass to just see if any vector gets in the neighborhood.
Yeah.
But I think that we as some of the vector goes out into the blood, and a decent amount of it does, then it circulates and it goes to all kinds of systemic tissues. So that's probably what's going on. But I have been kind of curious about, the mice will shed vector through their feces and urine, and mice, as they're cohabitated, I mean, they might ingest vector. I don't know.
Coprophagy is the pleasant word for that. Yeah.
Okay. Yeah.
Yeah, that's great talk.
I believe Dr. Schulze, Andreas, you have a question?
Yes, thank you. Thank you very much, Stephen. Gary Lipschitz raised a point, and I was thinking about the AAV9 brain delivery. In contrast to many of the conditions that you are working on that are neurodegenerative and fatal, we rather deal with neurodevelopmental disorders. And I think what we have learned is that early intervention is crucial. So ideally neonatal intervention. But when you think about gene therapy or gene delivery, and about the waning of the gene effect when you put this in a growing organ, what would you think about this? So would you still think about the concept of doing gene delivery in a neonate eventually?
Well, I mean, what we've seen is, I mean, I'll take CLN1, for example. If we treat mice early, like 1 week old, with a certain dose, we can get better efficacy than treating them at 2 months old with a 10 times higher dose. I mean, it's really the important, and that's a degenerative disease, but I think neurodevelopmental disorders, I think that the benefits that we might expect are just, it's going to depend on the biology of each disease. I think it's on a case-by-case basis. But I agree with you because I work on Rett syndrome. I work on SLC6A1, which is a GABA transporter. You know, in all of those, I would expect, and I think we've seen, early intervention is really important, even though those aren't degenerative. But I think in specific answer to your question about if you treat a growing brain, you know, and I don't think we're talking about 3 and 4 years old, maybe, because that's when, you know, the brain is kind of almost full grown at that point, but truly a newborn. It's an interesting question. I think that we've got an experiment ongoing with spinal muscular atrophy, where that's happening in the clinic across hundreds of patients, and eventually we'll have an answer to that. But
SMA is different because they are losing the motor neurons, right? So if they are lost, they are gone. That means you have either to treat them immediately or it will not work. I'm only thinking of, you know, as a family here, family members in this meeting right now. And these may be the people, the persons that we will approach to join clinical trials, you know? And now I'm thinking—
Sign us up.
Yeah, and this is not only, you know, this is not restricted to creatine deficiencies. We have the same thing with other metabolic diseases now, where we do have a treatment, but gene therapy would really be way, way better. But we have basically only one shot, kind of. And I don't know what to say to the parents.
Yeah, I don't know what to say either. You know, I spend a lot of my time trying to manage expectations, frankly.
I mean, here's what I'll say as a parent, right? I think we don't have a lot of options. So I think anything that moves science forward is a step in the right direction. And I know there are trade-offs and there are pluses and minuses, but I think, and we all have to analyze those risks, right, when it comes to it. But I think we're in a better situation with options than without them.
Yeah, and the other thing that I'll say is, like with CLN7, you know, the last disease I presented, it does not benefit from cross-correction. We're likely only targeting 5 to maybe 20% of cells across the brain, and we still saw like a really astounding benefit. So I think that this, you know, kind of brings up an interesting neuroscience, a neurodevelopmental kind of question about, you know, how can a mixture of healthy neurons and sick neurons, how does that actually manifest itself? And frankly, I think when we go into patients, when we do these clinical trials, I mean, everybody has to be aware that the clinical phase 1 clinical trial is not the finish line. This is the beginning of the real experiment.
So Steve, on that point, you spoke about managing expectations. I think there's one question in the QA box that would be relevant to addressing something like this, right? So you mentioned in your talk that gene therapy recipients have to take immune suppressive drugs for some of the time. Can you talk a little bit more about these kinds of alternate therapies that patients have to be on? What are some of the risks that patients have to be aware of when they're enrolling for a gene therapy clinical trial?
Well, so I'm not a clinician, so you know, I'm not giving like clinical advice or care advice, but you know, my opinion here is, you know, the same as you'd give anybody. I think you need to look very carefully at what the requirements are, look at very carefully at what the burden is going to be before you sign up for something like this, because participating in one of these clinical trials is, I mean, it's difficult. There's a lot of visits. Some of the things can be pretty invasive. You can be on immunosuppressant drugs that carry their own risks. But, you know, so every individual has to weigh their own personal situation and what the risks and benefits are to them as a, you know, personally.
So Steve, on a kind of tangential note, but what was the story of Hannah? What happened to Hannah?
So you can go to Hannah's Hope's website. So I guess I don't like to talk about individual participants in the trial, but you can go to Hannah's Hope's website, and they have a note on there that Hannah got enrolled in the trial, which they were very excited about a few years ago.
Okay, so I'll ask another question from our QA box. The talk that you just gave us focused on AAV9. Can you comment on some of the other AAV vectors? Specifically, how do AAV antibodies against naturally occurring viruses limit gene therapy efficacy? How much of a concern is this? Are there ways to circumvent this type of reactivity?
Yeah, you know, it doesn't get a lot of attention, but I think there's some data out there that AAV-RH10 might work to some extent, AAV7 might work to some extent. There's an emerging amount of synthetic capsids, like lab engineered capsids, that may even work better than AAV9. So this is a very fluid kind of situation. I mean, I'm hoping that we get to a point where we can get most of the cells in the brain with better vector technology. But I think the antibody situation is going to continue to be an issue because, you know, I mean, they're serotypes, so by definition, they're serologically distinct, but you still have a lot of cross-reactivity where, you know, maybe you have a high inhibition of AAV9, but then it's a lower inhibition of AAV8, you know, vice versa. So, you know, I think that was something that I really wanted to explore is if a patient was, you know, had antibodies against AAV9, can we just switch to a different capsid? But, you know, in reality, when we move to the intrathecal injection route, we haven't found anybody that has high enough levels of antibodies in the CSF to block dosing. And in terms of redosing, I think the antibody levels to AAV9 get so high that they're going to cross-react with probably most other capsids. You know, but it's a very hot, very heavy area of research to try to circumvent or deal with those anti-capsid immune responses.
So I'll pause for a minute and see if any of our panelists want to jump in with a quick question.
Oh, well, Stephen, I was going to ask, so in the world of transfusion medicine, sometimes what we do is if people have pre-existing antibodies, we'll do plasmapheresis to remove the antibodies. Is there any possibility that you could, if somebody had a pre-existing, you know, antibody that prevented them from being treated IV, you could plasmapherese it away and then get the virus in?
Well, I guess I'll put that back on you. I think if the antibody levels are low, and I've seen some data out there that yeah, you can. And so if you had an IV trial and you had somebody that maybe was just barely on the wrong end of the, you know, the antibody levels, then you might be able to plasmapheresis, get them down, dose them, and then, you know, move on. But if we have like a 1 to 10,000 antibody titer, I don't know if you're going to be able to plasmapheresis that away, if I could use that as a verb, you know, down to less than a 1 to 5 titer.
Yeah, no, that's true. It certainly matters with titer, absolutely. And whether it's IgM or IgG and yeah, that's tricky, but it's at least an option for patients who have higher titers.
Yeah.
Okay, so I'll go back to my QA box question. So there was a question on improving the targeting of AAV9 to brain cells. Can this be done without needing to increase the viral titer?
Yes. There's been a few publications now in mice where people have used different peptide insertion kind of library approaches, and they've pulled out variants of AAV9 that are 10 times, 100 times more efficient, you know, at the same dose. I think there's some of the early reports of those at least do not translate to primates. They only work in mice. And in fact, they only work in certain strains of mice. But I know the American Society of Cell and Gene Therapy meeting back in May, there were some presentations where people were, you know, a few different groups were presenting some early data that they might have capsids that are superior to AAV9 that work in monkeys. There was one presentation that, I mean, they presented something that was about 100 times more efficient than AAV9 in monkeys. So, you know, this is just, an emerging area. I think that the technology will get better. I think it will get much better. And we're going to hit a turning point where, we can go, these therapies are going to be a lot more effective due to better vector technology and safer.
That's great to hear. So AAV9 you mentioned, this is not a question from the box, my question, but AAV9 you mentioned is amenable at this time for a single dose gene therapy purely because of the immune response that it generates. If we were to use one of the other vectors, does that mean that multiple dosing or periodic dosing with those vectors is actually possible? And how does that relate to the half-life of the gene effect or the like? How do you measure something like that in the clinic?
So there are a number of strategies to try to enable redosing, but I haven't seen anything that is really concrete right now and sort of clinically implementable. Or at least there's nothing I've seen sort of a proof of concept about redosing in the clinic. So I think there's a lot of really smart people working on this. And I think that probably there will be some kind of options at some point, but right now we just go into everything assuming that it's a one and done. And in terms of the durability, if we're targeting neurons, neurons are extremely long-lived. I think that where you have the durability issues are more where people are looking at hepatic, like gene transfer to the liver or other other organs or cell types that kind of turn over gradually. But if we're targeting the nervous system, this is one of the reasons why it's a good target, is that your neurons should be, as long as you keep them healthy, they're there for life.
Okay, that's a promising note to end on. So we are a minute shy of, we ran over. Thank you for your time. But I thought that the Q&A session was really engaging. There was definitely a lot of questions from the participants, from the attendees as well. We've not gone through all of them. Just in the interest of time, I'm happy to direct these questions to you. And if you have the time to able to answer some of them, that would be great.